Hi, this video is going to cover exercise 2, parts 1 through 3 for the genetics lab of Bio 111 at Cal Poly SLO. This is mainly just all the exercises that have to do with blood types. Feel free to pause to complete the activities that I overview if I go over too fast. Always feel free to pause and rewind. This is being recorded by Ashley Fisher in the fall of 2021. So these exercises about blood types go over what a blood type actually is, and then knowing that information in blood transfusions, who can donate blood to who and who can receive blood from who based off of your blood type. And then we're also going to learn how to read a blood typing test. And if this is done in person or you are given the materials to do so, there also might be time to be able to determine your own blood type. If you don't even have the materials, we will still go over how to read the blood type test. All right, so quickly, let's just go over some basics. So red blood cells. This is what we use to determine blood type, because blood type refers to which, if any, microscopic sugars, or what we would refer to them as antigens, a person has attached to the surface of their red blood cells. So like I said, we call these things, these sugar tags, antigens. They're microscopic sugar markers or sugar tags that are attached to the outside of your blood cells. So they kind of like stick up microscopically all over the surface of each blood cell in your body. A different blood type signifies a different kind of sugar tag that your genes code for. So AB type blood does not have the same sugar tags that just B type or just A type blood has. So, like I mentioned, a genotype is what actually determines your phenotype or blood type that you could possibly have. So there are actually 4 varieties of these A, B, O type antigens that you can have, or phenotypes that you can have. So you can end up with type A blood, type B blood, type AB blood, and type O blood. And that's because there are 3 possible alleles you can inherit, which is what is indicated here with this ABO. So the type A allele codes for a specifically shaped sugar tag or A antigen, like we mentioned, and this is actually co-dominant with B. The B allele codes for a different sugar tag, and by different, I mean that they just have specific different shapes. And this different sugar tag we refer to as the B antigen or B sugar tag to red blood cell that is coded to be exhibited on the red blood cell surface. The type O allele, so this is not like a simple big D, little d, this is 3 different combinations. So the type O allele actually codes for no sugar tag at all to be on the red blood cell surface. So if someone has type O blood, they don't have any sugar tags on the surface that are A or B. They're just kind of naked. So the reason why we can have type AB type blood is because type A and type B alleles are co-dominant, which means that they can both be exhibited at the same time if you get one A from mom and one B from dad. We have type O listed essentially as recessive because if you get one O from mom and one A from dad, because you have an A antigen, you will exhibit those A alleles, which essentially makes you a type A blood type. Or those A antigens is what you will exhibit. So, these antigens can be really useful to protect yourself from invaders. So anything that is foreign in your body, we can consider an antigen. But if someone gets a blood transfusion and they don't have the same surface sugar tags that your blood actually has, so in this case, you can see that this would be someone's bloodstream where they have type A blood. If type B blood is put in there, those antigens can be recognized by your immune system. So the antigens that appear on your red blood cells tell your immune system which cells belong to you and which are foreign invaders that need to be attacked. So if you have type A blood, we can recognize our own type A blood by our own sugar tags. There's no foreign sugar tags that I wouldn't associate with. So any foreign molecule is what we would refer to as that foreign antigen in your blood, or these things can appear elsewhere in your body, and they can stimulate an innate or adaptive response by the immune system, which is generally really helpful in real life. You know, when we get bacteria, like if a bacteria thing floated into any part of our body, we want to make sure that that immune system actually responds to it appropriately. But it can become tricky when you are doing blood transfusions because you can have a bad response to the wrong blood type. So those arrows are just essentially pointing out the specific sugar tags on the antigens and the absence of them on the group O blood type. So if you've ever heard of someone saying I'm O positive or O negative, that's actually referring to another antigen we have. So our blood type is not just coded for by the ABO alleles that we inherit, it's also coded for another antigen that we call RH factors. Sometimes you'll see it as represented as a D elsewhere. We're going to use the RH terminology in this class. This is kind of like the O situation, where if you see a positive, that means you actually just have the RH factor allele coding for it to be present. So it's essentially either present or absent. People with the RH factor have a positive blood type. People without the RH factor have a negative blood type. This is like the O, like I mentioned, meaning that if you get one positive or present allele from your mom and you get a negative from your dad, you are still considered RH positive, because once you have one positive, that means you will exhibit that antigen, that RH antigen. So if you put 2 and 2 together of the different combinations of A, B, and O, and the different combinations of those with the positive or negative, you essentially have 8 total potential blood types that you could inherit, depending on the ABO antigens and the RH factor antigens. So for exercise 2, part 1, we would normally use whiteboards in a class setting, and you get into your table groups, and using different colors and this antigen drawing key, I'm going to have you guys draw out all possible red blood cell types on these whiteboards. So these you want to make sure to have in your lab notebook. If possible, definitely check with your instructor to make sure they're correct. And an example that I used using this key that is very sad because I drew it on the computer is showing an AB negative type blood cell. So you would put that in your table right here. As you can see, we have the antigen that is a little triangle that represents the A sugar tag or A antigen. We have the little B's in green for the B antigen, and RH is negative, or this is an AB negative person, which means we do not have the RH factor present. So there are no squigglies on this drawing. So you can go ahead and use this key to fill in the rest of this table in a lab notebook. I would pause the video here so you can go ahead and fill those out. And also you can use the internet to check your drawings as well, I'm sure, if you are doing this remotely. All right, so that was exercise 1, part 2. Now we're going to move on and talk about how the immune system reacts and what that has to do with blood transfusions. So like I mentioned, the immune system can recognize antigens that are foreign to its own body, and this causes clotting. So an antibody is a new term, and it refers to a protein produced by blood cells that can bind to a specific part of an antigen, tagging it for removal by the immune system. So essentially, you produce antibodies for anything foreign. They are represented like these little triangle things here, and you can see them here. These are binding sites. This is where they bind to anything foreign, and they are made for anything that is foreign in your body that your immune system wants to get rid of, to mark it for removal. So if you have A-type blood and someone comes in with B-type blood, these things would stick to that B sugar tag and tag it for removal. So the body produces these antibodies as a defense against the foreign proteins. In real life, they'd be in things like viruses or bacteria, and they hang out in your blood plasma. So not with your blood platelets, but they will be in your bloodstream if you're producing them to prevent foreign blood types going through. If you receive a blood transfusion from someone who has antigens that you don't have, your body will create antibodies, these little triangle hat things, against the foreign antigen, and it will cause severe clotting once this starts happening, which is what we call agglutination. So this is just another representation, like I mentioned before. If you have A-type blood, just ignoring whether there's Rh factors or not, and you get transfused with B-type blood, eventually your body produces these antibodies that recognize those foreign surface proteins, those B-shaped sugar tags, and they attack it like this, and it can cause essentially clotting or like a backup like this. And those clots can actually be really deadly if you imagine those traveling around your bloodstream. If they get caught anywhere, that's can be potentially deadly. So quickly to review, there are possible antigens that we have. So the A, the B, and the Rh antigens, which are those surface tags. And think about why remember if there's no antigen or antibody again for O types, think about absence versus presence. And then of course, the possible antibodies are A antibodies, B antibodies, and Rh antibodies. So essentially, you can create an antibody for any antigen or surface tag that is possibly present. Like I said, O is the absence of an antigen, right? So that would make sense why there would be no O antibody. All right. Blood transfusions. What does this have to do with blood transfusions? Well, patients can only accept blood transfusions of blood types against which they do not produce antibodies. So they can only accept they can only accept blood types that align with the surface proteins that they have. So we're going to go over which blood types you can donate to or accept in transfusions. So this is exercise 2, part 2, and this is that big transfusion compatibility table. So using whiteboards in the lab setting, you would take your group mates and you would fill in this table. Group 1 would do A's, group 2 would do B's, group 3 would do O's, and group 4 would do the AB type blood test. If you are alone, you can go ahead and do that all by yourself. The genotype sections are referring to the potential genotypes that would produce the specific blood type for each level. So those would be like something like AB, AO, OO, and then the RH genotype table column is referring to the RH alleles that you would inherit from your parents. So the genotypes possible could be plus plus, plus minus, or minus minus. And you have to match those with the appropriate blood types. And then you have columns for who you could potentially donate blood to, who you could potentially receive blood from. And then the last two columns are what antibodies you would produce to protect against foreign blood types if you given each blood type. And then the last column is the antibodies that would cause agglutination with the given blood type. So this is a table review. So once you do your best to fill out table 4.5, this will help as a reference. It's not the exact one, but it will still be helpful. I will include somewhere on PolyLearn, or your instructor will in future classes, a filled out table if you have not reviewed it or did not get a chance to review it in class. So once you fill out table 4.5 and you get comfortable with the difference between who makes what antibodies and who makes which antigens, we can go ahead and get into exercise 3, part 3, which is how to read a blood type test. So in this lab exercise, we're going to have you draw 4 images similar to this blood test, at least the blood test panel, with anti-A, anti-B, anti-RH, and a control. And at the end of this exercise, you're going to have 4 drawings total, and what it would look like for these blood types listed below, what their blood type test would look like. And so you'd have 4 drawings total. So I'm going to really quickly explain how this works. So what's happening here is that these are just clear panels where you can place a drop of blood from one person. So to figure out what kind of blood type you have, this is just a very clever way to do it. In a lab, people have figured out how to generate antibodies, like the kind that our immune system produces for B. So anti-B antibodies have been distilled. Anti-A antibodies have been distilled into vials that we have. And we also have anti-RH antibodies that have been distilled into serums. So we have these antibodies distilled into different serums. And so what happened here is, and then we also have a control, which is saline solution, which we'll discuss in a second. So what we did here essentially is, first, what you would do is you would put a drop of one person's blood, 1 drop each of the same person, into each of these little clear circles. So once you put that drop of blood into each of these circles, we will have special serums that have specifically anti-B, anti-A, or anti-RH. This is where we can add 1 drop of those antibody serums into each of their designated spots. And then we would take the toothpick and swirl these around and combine them. We also have a control. This is where we put 1 drop of blood with just saline solution. And this is essentially to make sure that there was no accidental contamination when we were mixing them up. Because if this control had clotting or agglutination, that would mean that this test was null and void and something got contaminated and you should start over. But basically what's happening is it's going to be either clear, which means there is no clumping or no agglutination, or you're going to see clotting like you see in the anti-RH and the anti-A sections of this test. That means that this person's drop of blood, when it mixed with the anti-A antibodies, those antibodies descended upon the A sugar tags that were present in this person's blood. So if they didn't have if they did not have A type blood or those A sugar tags, it would look just like the control or just like the anti-B, it would be clear. But this person actually has A sugar tags and RH sugar tags, which indicates that this person has A positive blood. So that would be the drawing you would draw for number 1. You can take these concepts and essentially apply that to 2, 3, and 4 of your drawings. And if you have these materials with you, or you're going into lab to do this at some point, you will be given a placemat, a lancet, an alcohol wipe, a toothpick, which we're going to break into 2 pieces each. So you'd have essentially 4 little stir sticks, saline to add to the control circle, a Band-Aid to cover your pricked fingers, and your antibody serums in different vials. So anti-A, anti-RH, and anti-B serums of those antibodies. You can use these materials essentially to carry out the test, just as I described. 1 toothpick should be used for each of these separately. If you mix toothpicks up again, you could potentially contaminate these, and it might say, holy mackerel, you have AB positive blood, when in reality you just have B negative blood, but you used a toothpick that accidentally contaminated the other circles. This is about cleanup. You can pause to read that more. Your instructor will tell you about it in person. These are more directions. And this is table 4.5 to review later. Go ahead and pause this video if you'd like to spend more time with it. All right, so I'm going to talk you through this blood typing kit that someone is demonstrating here on YouTube, just to hammer home how we determine this blood type. So what you can see here is the solution with the antibodies for anti-A, anti-B, anti-D, and the control have already been applied. They clearly put coloring in them so you can tell them apart, and they are now dissolved in that solution. So if you add blood to them, they will show that agglutination or clumping if those applicable sugar tags are present in the blood that is being applied. Warning, blood is being shown. So 1 drop of blood, like they mentioned, from the same sample, same blood type, same person, is being added to each of the antibody solutions. Now what the person is doing is combining the control saline solution with the blood to make sure that there is no agglutination happening, and this test is valid. Now they're mixing the anti-D in, so that's testing for the presence of the RH sugar tag. They go ahead and mix anti-B. Important to note that they are using different mixers, separate mixers for each different sample. And that's, of course, because you want to avoid cross-contamination. So what you can see here already, even though they're setting a timer, is that there is definitely some agglutination going on. Anti-A and anti-B, those antibodies are definitely clumping onto the blood, which indicates that there are presence of those sugar tags in this blood type. Even anti-D is kind of clotting a little bit too. So I want you to think about what blood type this person would have, since all 3 of them are clotting up: anti-D, anti-B, and anti-A. All right, that is it for this recording. That should be the total overview of blood types and all the exercises associated from the genetics lab for Bio 111 at Cal Poly San Luis Obispo. Definitely email your instructors or go to office hours if you have any questions.